Biological monitoring is the collection of biological data, specifically macroinvertebrate data. In this chapter, we will cover how to collect this data and how it helps us interpret stream health. The topics covered in this chapter are an introduction to biological monitoring, identification of benthic macroinvertebrates, how to collect the macroinvertebrates, and how to record and interpret the data. To start things off, we'll define benthic macroinvertebrates, the advantages of using biological sampling as an indicator of water quality, and the different categories of pollution tolerances that categorize different species of macroinvertebrates. It's easiest to break down the term benthic macroinvertebrates into smaller parts to understand what it means. Benthic refers to the bottom of the stream. Macro means large or visible with the naked eye. Invertebrates are organisms without a backbone. So benthic macroinvertebrates are organisms that lack a backbone, are visible without magnification, and live on the stream bottom. An example is a crayfish or crawdad. These invertebrates play a vital role in the stream ecosystem. They are very important to the food chain, providing energy for numerous fish and other species. They also influence the nutrient cycle, primary productivity, decomposition of organic matter, and moving small materials within a stream system. Invertebrates are indicators of stream health. There are several reasons why it's advantageous to use these spineless critters in assessing water quality. Invertebrates are relatively non-mobile, whereas fish will move if conditions start to deteriorate. They are also easy to collect. We just need a net and something to agitate the rocks, like our 2 feet. Some invertebrate species are sensitive to pollution, while others are more tolerant. This allows us to determine the condition of a stream based on the presence or absence of certain taxa. Sampling macroinvertebrates allows for continuous monitoring. They are permanent residents of the stream. This makes them susceptible to pollutants present in the water. Chemical sampling only tells you what is happening in the stream at the exact moment you collect the sample. Some pollutants pulse through a stream due to variations in flow or to discharge from point sources that are released at intermittent times. The equipment needed to collect macroinvertebrates is inexpensive compared to chemical monitoring equipment. While debatable, macroinvertebrates are easy to ID. It may be difficult at first, but with practice, common invertebrates from your stream become familiar and easy to identify. Finally, chemicals are not needed for macroinvertebrate sampling. This eliminates handling of chemicals, the need for reagent disposal, and replacement of expired chemicals. Most invertebrates collected for biological monitoring are insect larvae or nymphs. The taxonomic level they are identified down to depends on the ability of distinguishing the organisms with the bare eye, and maybe the help of a small magnifier. Taxonomic classification is a hierarchical classification of organisms, with kingdom being the broadest and species being the most specific and most difficult to distinguish. You won't need to know the taxonomic levels for water quality monitoring. This just shows us how close some of the organisms are related, and that some may have very similar characteristics. We also define the tolerance levels to the taxonomic levels for which we ID. For example, we will generally state that caddisflies are sensitive to pollution, but some species of caddisfly are more tolerant to pollution. Let's talk a little more on pollution tolerance, since this helps us understand how the invertebrates present indicate the health of the stream. We consider 3 levels of pollution tolerance: pollution sensitive, somewhat pollution tolerant, and pollution tolerant. Invertebrates that are pollution sensitive will only be present in streams with excellent water quality. Pollution tolerant invertebrates can be found in streams with severe impairment. Note that pollution tolerant organisms can be found in all streams, including those of excellent water quality. We hope to find a diverse community with invertebrates from all 3 categories. This image shows a fish fly larva, which is somewhat tolerant to pollution. The Missouri Stream Team program often references the Stream Insects and Crustaceans Identification Resource. This is also referred to as the Blue Bug Card because it's printed on light blue paper. The Blue Bug Card was adapted from the invertebrate identification tool produced by Isaac Walton League in Maryland. It is used commonly by water quality monitors from many states. This resource shows invertebrates taxa by pollution sensitivity. Listed under Group 1 is pollution sensitive. Group 2 shows somewhat pollution tolerant. Group 3 is pollution tolerant and can be found on the back side. There are size bars located next to the taxa of invertebrates. The full bar shows the maximum size, and the black portion of the bar shows the average size. Here is the backside of the blue bug card, showing the remaining Group 2 taxa and Group 3 taxa. You may notice that the sensitive organisms in Group 1 have legs and gills, while the pollution-tolerant organisms in Group 3 lack legs and gills. The blue bug card can be found on the Missouri Stream Team website under Water Quality Monitoring, Training Material and Resources, or a free copy print can be requested through Stream Team staff. Part of biological monitoring is identifying the invertebrates collected. We will give tips on identifying each taxa. This information can be found in the introductory level notebook under Water Quality Monitoring Resources on the website. Let's jump into identification of pollution-sensitive invertebrates. The pollution-sensitive macroinvertebrates are located on the front of the blue bug card. We will work through each taxa in no particular order, explaining key characteristics and tips for identification. For many of the invertebrates collected, they will have a head, thorax, and abdomen. It's important to be able to identify these three body parts. This drawing is a mayfly. You will see it has 3 pairs of legs. Most mayflies also have 3 tails. Some mayflies only have 2 tails, and some mayflies may lose tails while being collected. So the number of tails can be an identifying clue, but don't rely on it solely. In this drawing, you can see a row of plate-like gills along the abdomen. This is a key characteristic for identifying mayflies. Remember, mayflies usually have 3 tails and gills along the abdomen. Here is a photo of a mayfly nymph. There are gills along the abdomen. When placed in a sorting tray with water, you can often see the gills undulating as the mayfly is moving water across the gills to breathe. This species also has 3 hair-like tails. This is also a mayfly, as shown by the gills on the abdomen. It looks different from the last mayfly image because they are 2 different species of mayflies. Again, look for the gills on the abdomen and 3 tails to identify a mayfly. This is a stonefly nymph. It is the most pollution-sensitive macroinvertebrate you will collect. It has a head, thorax, and abdomen. There are no gills on the abdomen. Instead, the hair-like gills are on the thorax under the legs. The stonefly has 2 tails. The key identifying characteristics are the 2 tails and gills on the thorax. Here is a picture of a stonefly nymph. Notice the gills on the thorax under the legs. When placed in a sorting tray of water, you may see the stonefly doing push-ups to move water across the gills. Here is another species of stonefly. The gills are not as easy to see, but we can see that it has 2 tails. This is another photo of the giant stonefly, which is the largest stonefly in Missouri. Stoneflies tend to be larger and more robust than many other invertebrates. This invertebrate is quite a bit different than the mayflies and stoneflies. The caddisfly larva still has a head, thorax, abdomen, and 3 pairs of legs. There are gills along the abdomen. Caddisfly do not have tails. Instead, they have hooks called prolegs. Along the thorax are 1 to 3 hard chitinous plates. Some species of caddisfly are free-living. Some are web-spinning, yes, like spiders. And some are case-makers. Here is a picture of a free-living caddisfly. Notice the hard plates on the thorax, 6 legs, and the soft abdomen. Gills are also observable along the abdomen. There are also 2 prolegs, which should not be confused with tails. Here is another image of a common caddisfly. Sometimes they can be yellow in color or green. The image on the left shows a net-spinning caddisfly. The spinneret of a caddisfly is located under its head. The photo on the right shows a cased caddisfly larva. Cased caddisflies build their cases using silk and material of preference. Species can actually be identified by their case materials and shape, but that is not necessary for water quality monitoring. This is another picture of a caddisfly in a case made from detritus or leaf material. Anytime you find gravel stuck together or a tube of rocks or leaves, it is a strong indication of the presence of caddisflies. This image is not a caddisfly case you will find in Missouri streams. Jewelry makers have used the assistance of caddisflies to make unique necklaces and earrings. After a caddisfly pupates, it will leave behind the case for the jeweler to create accessories. Most of what you collect for biological monitoring will be juveniles, larvae, and nymphs. The riffle beetle is an exception. You will collect both the juveniles and the adults of this aquatic beetle. The image on the left is the juvenile. As you can see, it is very small. The adult is equally tiny. Riffle beetle larvae have a head, thorax, and abdomen, and 3 pairs of legs. The thorax and abdomen are hard. The larvae have gills, but they are usually covered by a plate at the tip of the abdomen. The adult riffle beetle is small and dark in color. Both adults and larvae will be counted together for the data sheet, which will be covered later in the chapter. Another beetle collected may be the water penny. This is a juvenile or larval beetle. Water pennies have been described as looking like a fish scale stuck on a rock. This can commonly be found clinging to the bottom of rocks in the stream. The image on the left shows the top or dorsal side of the water penny larva. Looking at the underside of the water penny larva, you can see the head, 3 pairs of legs, and gills. The only pollution-sensitive taxa collected that does not have legs is the gilled snail. We also refer to the gilled snail as a right-handed snail because the shell opens to the right when held with the apex or point facing up. The gilled snail has an operculum which looks like a trap door over the opening. Empty shells are not counted in biological monitoring. We only count live organisms. This image shows the average size of gilled snails. Note that the shell opens to the right when the apex is pointing up. Aquatic snails can often be observed by just looking into a stream along the gravel or plants. As you are learning to identify invertebrates, you may try using different identification resources, like a dichotomous key. A dichotomous key gives 2 choices for features until you finally land on an ID. Let's practice using a dichotomous key for this invertebrate. The dichotomous key can be found on the website and can also be requested as a free print through Stream Team staff. Starting at the top of the key, you will decide between the 2 choices which one best describes the invertebrates. This invertebrate does not have a shell. It has 3 pairs of legs, no wings. It has 1 or 2 tails. From these final choices of features, we can see that it has large mouth parts and spines on its side. This invertebrate is a dobson fly larva, also called a hellgrammite. Hellgrammites, the larval stage of the dobson fly, are the largest invertebrates which can be 4 inches in length. These can have 2 to 3 year larval period, so if you find a large hellgrammite, it can be an indication that no major pollution events have occurred in your stream for a long time. Key identifying characteristics of hellgrammites include large mandibles, filaments along the side of the abdomen, and gill tufts under the filaments. The color in the head may also appear mottled compared to other similar taxa. When placed in a sorting tray of water, the gill tufts can be seen. When stressed, the hellgrammite may roll into a ball and the gills will be concealed. Next, we will discuss macroinvertebrates that are somewhat tolerant to pollution. If you are referencing the blue bug card, we will be working through Group 2 taxa. The crayfish is one of the most easily recognized macroinvertebrates. There are over 30 species of crayfish in Missouri, 7 of which are found nowhere else in the world, and another 12 that share only limited range with neighboring states. If you find a crayfish in your net, remove them immediately, record the number on your data sheet, and return them to the water. Otherwise, they may escape or make a meal of the other captured invertebrates, resulting in an inaccurate biological count. This is a sowbug, or isopod. It is a crustacean similar to the crayfish. It is easy to identify because it resembles its terrestrial cousin, the roly-poly or pillbug. The sowbug has several pairs of legs and appears flattened horizontally. The scud, also has several pairs of legs, but appears to be flattened vertically. This can be confused with freshwater shrimp, but it is smaller in size. When placed in a sorting tray of water, the scud swims quickly and often on its side. For this, it is also referred to as a side swimmer. Here is another photo of a scud, showing the several pairs of legs. The alderfly larva is similar in appearance to the hellgrammite. They have the same pinching mouthparts. The most obvious difference is that the alderfly does not have gills under its filaments. The filaments on the alderfly have a much more delicate appearance than those of a hellgrammite. The alderfly is also much smaller than the hellgrammite. The abdomen ends in a point that looks like a tail in the shape of the capital letter A. We can say it's A for alderfly. This image is a fish fly larva. It looks almost identical to the hellgrammite, except it does not have gills under the lateral filaments. The fish fly is also smaller in relative size than the hellgrammite. But remember, some hellgrammites can be smaller and easily confused with fish fly. The key identifying characteristic is the lack of gills under the lateral filaments. While not used as an identifying feature, breathing tubes may be seen on fish flies under a magnification. The damselfly nymph has 3 broad oar-shaped gills at the end of its abdomen. These should not be confused with the 3 hair-like tails of mayflies. Like tails, the gills can be easily broken off while collecting invertebrates. When placed in a sorting tray with water, the damselfly often stands with the gills pointed upwards. Damselflies and dragonflies are usually found along the slower moving water along the stream banks and vegetation. Here are 2 other species of damselflies. While the body length differs, the oar-like shape of the gills are similar. Dragonfly nymphs are closely related to the damselfly. They are relatively large in size and lack a tail and gills. The abdomen is relatively wide and has a leaf-like appearance. Like the damselfly, the dragonfly also has a long folded lower labium used for capturing prey. Dragonfly nymphs can vary in size and shape. These are a couple smaller species of dragonfly. Here are drawings that show other shapes of dragonfly nymphs. The key identifying characteristics is the lack of tail and gills and the leaf-like shape of the abdomen. Water snipe flies are true flies with aquatic larvae. They have body segments with prolegs on each. Another identifying characteristic is the 2 feathery horns. Water snipe flies are very close in appearance to the common horsefly or deerfly. They are about the same size and cream color. Remember, the water snipe fly has caterpillar-like prolegs on each body segment and 2 feathery horns. The horsefly is pointed at both ends and has creeping welts around each body segment. Horseflies are not counted in biological monitoring, but they can be recorded in the comment section of the datasheet. Cranefly larvae are another aquatic true fly larvae. Most species are large, about the size of an index finger, and squishy. They have finger-like projections or tulip-like bulb at the posterior end, which helps them anchor into substrate. When placed in a sorting tray with water, cranefly larvae can be very active and escape the tray. Occasionally, monitors may find larvae of other aquatic beetles, other than riffle beetles or water pennies. Other beetle larvae can be identified by process of elimination. If the characteristics don't quite match those of other taxa, it's probably other beetle larvae. If you are uncertain of an ID, you can take a photo and email it to Stream Team staff to help with identification. Freshwater mussels are somewhat tolerant to pollution. This image shows 2 native mussels, the maple leaf. Native mussels tend to be asymmetrical and large compared to the invasive Asiatic clam. It is estimated that over 70% of native mussels are either threatened or endangered. Therefore, volunteers are encouraged to return all clams and mussels to the stream system as quickly as possible in case they have collected one of the many threatened species. Mussels must be placed upright in the substrate with the umbo or hinge up. Like snails, do not count empty shells on the datasheet, We only count live organisms. Finally, we will discuss macroinvertebrates that are tolerant to pollution. If you are following along with the blue bug card, we will now go over Group 3 taxa. As a reminder, pollution-tolerant taxa can be found in all waters, polluted or healthy. The presence of a pollution-tolerant species does not indicate impaired water. Lack of diversity and/or pollution-sensitive taxa indicates impaired water. Aquatic earthworms are easily identified because they look like their terrestrial counterparts. They are segmented, long, thin, and often curl back around on themselves. Note on both the blue bug card and on your data sheets, all worm-like organisms, such as aquatic earthworms and horsehair worms, are lumped into one category called aquatic worms. Midgefly larvae are very tolerant to pollution. They are tiny in size. Midge larvae do not have legs, but do have a proleg under their head, which can be seen on larger specimens. Midge larvae can be red in color. This type of midge is commonly called a bloodworm, which is a misnomer because they are fly larvae, not worms. This type of midge carries hemoglobin, which provides them a source of oxygen so they can live in water with very low oxygen levels, such as swamps. The black fly larvae have a wide posterior, making it a bowling pin shape. They are very small in size. The posterior has small hooks, which allows it to adhere to surfaces with a little silk. When placed in a sorting tray with water, the black fly larvae often stick to the sides of the tray and sway in the water. Here is an image showing the filter feeding features of the black fly larvae. Again, these are very small. Likely the only characteristics you will be able to see is the behavior of the larvae sticking to the sides of the sorting tray. Common leeches in Missouri can look like a heavily muscled worm that is brown or black or mottled in color. The major identifying characteristics of the leech is the presence of a section at one or both ends of the ventral or underside. Monitors often confuse a planarian or flatworm with a leech. The planarian has a tapered head and two eye spots which may be faint but still present. They average about ½ an inch, while a leech can grow to several inches. Planarian are not counted on the datasheet but can still be included in the comments field at the bottom. Planarian can fragment and regenerate, so you may have 4 planarian in your net, but they may all be from one single organism. For this reason, they are not included in the count. A pouch snail lacks gills and an operculum, or trapdoor that covers the opening. It is also referred to as a left-handed snail since the shell opens to the left when the apex is upwards. Not all snails you find will be left or right conical snails. Some snails are coiled or look like ram's horns. These go in the Other Snails category on the datasheet. Many monitors will collect invertebrates for biological monitoring, then return them to the stream immediately after. Some monitors may choose to keep invertebrates for a specimen collection or assistance with identification. We will go over how to properly preserve invertebrates that are kept in a specimen collection. It is recommended to euthanize invertebrates in a 100% alcohol solution. After at least 24 hours, the specimens may be transferred to a storage solution. Ethyl alcohol or ethanol is a preferred alcohol as it is less harsh on the specimens. Isopropyl alcohol may also be used for preserving specimens. This is found in most households. Be aware of the concentration listed on the bottle to know how much water to add for diluting to 40% solution. Hand sanitizer may be used for small specimens like black fly larvae and midge larvae. This medium allows for easier viewing of the specimens. Before you can identify macroinvertebrates in your stream, you will need to collect them. We'll go over the methods to collect invertebrates from the stream. The first step in biological monitoring is collecting the invertebrates. Next, you will identify the invertebrates, then count them. The taxa present and quantities will help you analyze the stream's health. The Missouri Stream Team program provides a kick net, forceps, and magnifying lenses for biological monitoring. A monitor will need to supply their own sorting tray. It's recommended to use 1 or 2 white ice cube trays to easily sort and view invertebrates. A squirt bottle is also helpful for warm or sunny days to help keep the net wet while picking invertebrates. To sample for macroinvertebrates, you'll need to find 3 different microhabitats in your stream for your 3 net sets. These microhabitats can be 3 different areas of riffle, like in the photo, or combinations of different habitats if there's not enough riffle habitat in your site for 3 samples. Biological monitoring is conducted twice per year, once in the late winter, early spring before leaf on, and once in the late summer, early autumn before the leaves fall. This is because aquatic and macroinvertebrates are largest at these times of year and easiest to identify. Missouri has 5 aquatic faunal regions: Big River region, Lowland region, Ozark region, Glaciated Plains region, and Osage Plains region. These last 2 are combined and referred to as the Prairie region. Each of the regions are characterized by different habitats and animals. For example, riffles are common in the Ozark region, but are not common in prairie regions due to the gradient of the land. In the Ozark region, riffles and root mats are a common aquatic habitat. Prairie and lowland regions characteristically have root mats, snags, and pools. These habitats are listed in order of preference for sampling. Missouri Stream Team protocols are set up to sample riffles, but if your site does not contain a riffle, you can alternatively sample root mat. If there are no root mats, you can sample snags. And finally, the last resort is sampling from a pool. We will discuss each habitat in detail and how to sample within each, starting with riffles. A riffle is a section of stream where water breaks over rocks. This action incorporates atmospheric oxygen into the water, resulting in higher dissolved oxygen levels. This means more oxygen for invertebrates. Riffle habitat provides a variety of microhabitats for a diverse community of organisms. If this was our monitoring site, we could sample 3 microhabitats from these riffles, including the downstream riffle, the upstream riffle, and the riffle along the vegetation. When sampling macroinvertebrates, start at the most downstream part of your site and work your way upstream. This prevents disturbing the other locations you will be sampling. These are the steps to sample invertebrates from a riffle habitat. Essentially, you'll be placing a net in the stream and dislodging invertebrates so they float into the net. First, place the program-provided kick net in the riffle. Ensure the bottom of the net is taut and against the stream bottom. Medium to large-sized rocks can be used to weigh down the front of the net. If there are any large rocks or boulders in the 3-foot area in front of the net, you can pick these up and rub them over the net before setting them aside. This will ensure you capture the invertebrates clinging to the rock while keeping your feet safe for the next step. In the 3 feet by 3 feet area in front of the net, twist your feet and agitate the substrate. The goal is to dislodge invertebrates into the water column so they float into the net. After you are done agitating the stream bottom, rub and remove the rocks from the front of the net. Slowly lift the net from the stream, taking care to not spill the water and invertebrates from the net. Carefully move the net to the stream bank. Now is the fun part of picking invertebrates from the net and placing them in the sorting tray. Recording the data from this net set will be covered later in the chapter. If you don't have any riffles within your monitoring site, the next habitat to look for is root mat. Root mats are the fibrous roots that hang out of or over a stream bank and into the water. Sampling in different habitats will reveal different communities of invertebrates. Damselflies and dragonflies are commonly found in root mats in vegetation. To sample invertebrates from a root mat, the same program-provided kick net is used. First, place the kick net downstream of the root mat with the edge next to the bank. Kick and swirl the water under the root mat so invertebrates dislodge and float down into the net. Use your foot to disturb the root mat, not your hands. There may be biting critters in there, or poison ivy. Slowly lift the net from the stream, making sure that water and invertebrates do not spill over the side. Move the net to the land to pick and sort the invertebrates into a sorting tray. If your site has no riffles or root mats to sample, you can sample snags or woody debris. A snag is a tree, a limb, or other woody debris in the water. Once the debris begins to decompose, it becomes soft and invertebrates start to move in. To sample a snag or woody debris, place the net horizontally under the woody debris. Use a stiff bristled brush to scrub the surface of the snag. The goal is to dislodge the invertebrates so they fall or float into the net. Do not use your bare hands. Move the net to land and pick and sort invertebrates into a sorting tray. 3 to 4 pieces of small woody debris, like the one in this photo, are needed for a single net set. If no other habitats exist in your monitoring site, you can sample in pools or non-flow areas. When sampling in a pool, the flow of water is improvised by shuffling backwards and trailing the D net in front of you to catch the invertebrates. A D-frame net is required if sampling non-flow areas. First, hold the net downstream of where you are standing with the opening of the net facing you. Shuffle your feet to agitate the substrate. While shuffling your feet, sweep the net back and forth to capture suspended invertebrates. Continue shuffling and sweeping until you have sampled a 3 feet by 3 feet area. Remove the net from the water and take to land to sort the invertebrates. For any habitat sampled, always collect invertebrates in an upstream direction. Start at the riffle or habitat furthest downstream within your site, then work upstream. Avoid walking through habitat that you plan to sample. Disturbing the habitat before you sample can dislodge the invertebrates before you're ready to collect them. Be consistent in the habitat you sample. Aim for sample replicates of the same habitat each time you monitor. Ideally, this would be 3 riffles each monitoring trip. After invertebrates are collected and identified, how do you record the data? We will cover data recording and analysis next. After invertebrates are collected from the stream, you will remove them from the net, sort them into trays, identify them, and count them to record on the data sheet. If fish are collected in the net, return them to the water immediately. If any crayfish or mussels are collected in the net, count them and immediately return them to the water. Let's take a look at the macroinvertebrate data sheet, also referred to as a biological monitoring data sheet. As with every data sheet, make sure the header is filled out entirely. 3 net sets are required for this data to be accepted. This means you'll take 3 trips into the water to collect invertebrates, sort them, ID them, and count them. For each of these net sets, you will record the habitat type and select the type of net you used, which is a kick net for most monitors. The amount of time spent picking invertebrates off the net is recorded, but we do not record the amount of time identifying. We also record the number of people that help pick the net. For each invertebrate taxa, record the number for the respective net set. This needs to be a number, not a check for presence and absence. In the far right column, it shows circle types present and a corresponding weight or score for each taxa. The sensitive taxa have a score of 3, while tolerant taxa are a 1. If there is a presence of a taxa in any of the 3 net sets, circle the corresponding score in the right column. These will add up to give the water quality rating. A score of less than 12 can indicate poor water quality conditions. 12 to 17 is fair, 18 to 23 is good, and greater than 23 indicates excellent water quality. Instructions and tips can be found on the back side of the data sheet if you feel lost. Let's look at an example of why it's important to compare data over time. This data was collected on April 6, 2008 on Turkey Creek. There is good diversity of organisms found, and the sensitive taxa are plentiful. The water quality rating is 25, which indicates excellent water quality. Exactly 1 year later, another datasheet was completed on April 6, 2009, at the same site on Turkey Creek. Notice there has been a shift in populations. There are now more tolerant taxa than sensitive, but the water quality rating is still 25. Something has happened in the stream system to shift the populations. One of the other data sheets, such as chemistry or visual survey, may provide some insight to what has changed to cause the shift. This example also shows why it's important to record quantity, not just presence-absence. There are many factors that can affect water quality rating, which may not necessarily reflect onto the stream health. The type of habitat the stream provides will affect the rating. Silt and sand bottom streams will generally have lower ratings than streams with cobble substrate. Sensitive organisms require water with some flow because it helps oxygenate the water, but too much velocity can dislodge and sweep away invertebrates. Invertebrates thrive in cooler water where oxygen is also higher. It may be difficult to find invertebrates in a warm stream in the hot summer sun or where there is not shade from a riparian corridor. Different invertebrate communities can be found in different stream sizes, like small headwater streams or larger third-order streams. Aquatic habitats and stream flow are very different between these two sizes, therefore support different communities. A balance of chemical constituents must be maintained to support a diversity of aquatic life. Imbalances will result in changes in the stream that will alter what can live there. Certain chemicals are toxic to organisms, and if they are present in large enough quantities or for a long enough time, it will kill organisms in the stream. Habitat, flow, and rates of soil erosion are all physical factors that affect aquatic life. Poor ratings can often attribute to physical conditions in the stream rather than a point source discharge or single chemical pollutant. Finally, the taxonomic level to which we identify can also have an effect on water quality rating. We generalize pollution tolerance to an entire taxa, but in reality, some species under that taxa may be more or less tolerant to pollution. After you collect macroinvertebrate data, you may want to know how to analyze your findings. Let's go over a few scenarios which monitors may experience. If your monitoring site has a high density or a lot of invertebrates, a high diversity or many different taxa of invertebrates, including many sensitive, this would indicate the stream is healthy and water quality is good. In this scenario, we still have high diversity, but we have low density of invertebrates. So lots of different kinds of invertebrates, just not very many of anything. This could indicate poor habitat conditions or recent flooding. In this third scenario, we have high density but low diversity. So lots of invertebrates, but only a few different taxa. This could indicate too many nutrients in the stream or eutrophication. You may observe sedimentation or an algal bloom. In this last scenario, we have very few invertebrates and not very many taxa. The water appears clear and clean. This could indicate toxic pollution, such as chlorine. This can occur if there is a water main break or if a pool is draining into the stream. While we have identified lots of invertebrates you will be identifying in the field, there are some other organisms to consider, from endangered species to nuisance species. There is an endangered species that spawns in riffles during times when monitors like to sample streams. The Nyangwa darter is a small fish that is listed as Missouri state endangered and federally threatened. Because the Nyangwa darter spawns in riffles, kicking up macroinvertebrates can be detrimental to spawning. For monitoring sites in the Nyangwa darter range, we ask that monitors avoid sampling between March 15th through June 15th. Here is a map of Nyangwa darter range. This includes the Nyangwa River watershed, the Sac River watershed, the Tavern Creek watershed, and the Marys River and Pomme de Terre River. Again, please do not monitor the streams between March 15th and June 15th. It's important to be mindful of nuisance species as well. Some of the invasive species in Missouri streams are zebra mussel, Chinese mystery snail, rusty crayfish, and hydrilla. Familiarize yourself with possible nuisance species in your streams. It's important to always clean and dry your equipment, boots, and boats after being in the water to prevent spreading these species. Most monitors are not sampling in multiple streams on the same day, so their equipment is dry by the time they sample again. But if you are sampling in multiple streams in a single day, you will need to disinfect your equipment. Here are a few methods to disinfect monitoring equipment and waders. The easiest method is air drying all equipment for 3 to 5 days. If a monitor is going between bodies of water more frequently, vinegar or bleach may be used. This table can be found in the Introduction to Volunteer Water Quality Monitoring Notebook on the Stream Team website. If you want to test your knowledge on biological monitoring, you can pause this video and review these questions. Additional resources and training videos can be found online. Contact us if you have any questions.